Good morning, everybody. Welcome to our showcase for this quarter, and I'll let Nicola introduce and tell you all about what we're going to talk about and about drug substance and everything else as well.
Thank you very much, Paul. Good morning, good evening to you, depending where you are in the world, and welcome to our second drug substance showcase. We have got a nice group of our work streams presenting to you today, but just before we get into that, a little introduction of myself and of the Drug Substance Forum. So I'm Nicola Powell, I'm Senior Account Manager with the Drug Substance Forum. For any of you, we know we've got a mixed audience here today, so we've got members, people that are well familiar with drug substance and with BioPhorum. We've also got people perhaps meeting us for the first time, so welcome to you all. BioPhorum, if this is the first time that you are meeting us, is a unique collaboration of the biopharmaceutical industry. We are able to bring together biomanufacturers, CDMOs, suppliers to work together to solve some of the biggest challenges in our industry. And in BioPhorum, we organise ourselves into a number of different forums focusing on different areas of the industry. So the team that is representing here today is from the Drug Substance Forum. We've put together this series of webinars just to introduce the work of our work streams, let you know what we are working on, what we're working to deliver, and also hopefully give you a bit of a chance to know if this might be something of interest for you to join, or at least be aware of what upcoming deliverables, publications we're producing, so you are able to track our progress. So for the session today, a little bit of housekeeping. So we have got everyone joining, hopefully with microphones and cameras enabled. We will ask you while the presenters are presenting just to go off camera, mute your mics, because we are expecting quite a few to join the call, but we do welcome you to please, hopefully reasonably interactive session. So if you have questions, please feel free, raise your hand, you can turn microphone on, turn on camera. We love seeing you and hearing from you. It is just a high-level introduction today, but as I say, we do hope it will be quite informal and interactive. Not sure how many people join a webinar to be interactive, but we'll see what we can do. I think Paul has some very exciting polls for us, so there's at least a little bit of interaction there, but questions are very, very welcome. And we will make the slides and recording available after the call. On each of the sections, you'll see it has our contact details. We are here to help, so please do not hesitate to contact us if we can provide any further information. So just one more slide from me before we get into the interesting part, just to give you a quick overview of the Drug Substance Forum. So what we have on the screen now is just the current drug substance work streams and sub-teams. So across the top row here, you can see our main work streams, the different areas that we have SME teams focusing on a number of deliverables. And depending on the size of that work stream, the number of different focus areas, we may then also organise ourselves into sub-teams within that work stream to tackle different topics or different communications of our work. So each of these work streams will feature in one of our showcase sessions for the year. So these are quarterly sessions. If you're seeing anything on the screen that you'd really like to also see that one, then we do have details on the upcoming sessions towards the end of the deck. If it's one that you've missed, we do have slides and recordings. So once again, just let us know. But with that, I think I will hand over to Paul, who is going to present on our first workstream of the day, which is closed systems. Thank you very much, Paul.
No worries, thank you. I'll just get my slides. Here we go. So yes, closed systems. I'm going to talk to you about closed systems today. We've also got microbial control and Annex 1 applied in drug substance. So we've got 3 work streams that are very closely linked, but it's fortunate as well in some ways in that these show the different sorts of work streams that we've got as well, in that we've got closed systems, which is very much a transformation work stream. We're trying to change the industry and have been doing that for a period of time. And we've got microbial control, which is more of an operationals focused work stream, and then Annex 1 applied in DS, which is more of a reactive work stream. We're reacting to something that's happened with the publication of Annex 1. Our work streams always have a charter, and this is our closed systems charter. I won't go into too much detail because I'm going to talk all about the things that are on there. But the one thing that I would mention is closed systems do seem to be getting to the point where they're accepted in the industry. When we first started this work stream off, and it's one of the oldest work streams within BioPhorum. I think it started back in 2011, 2010, back then, and closed systems weren't accepted at that point, and they've become so they are accepted now. But we're still seeing them operating into a higher classification room, and that's the purpose of this workstream, is to try and get us so that we can operate closed systems in a lower classified room as possible. And we say CNC. We've had some debate about that and whether it should just be in a non-classified space, because CNC is up to us how we all define it. So I'll try and avoid the word CNC as we go forward. So this all started, and the reason that this all started was when Q7 was published by ICH, and it had this statement in there that I've pulled out at the bottom here that said, when the equipment is closed, you can operate the equipment outside, because it provides an adequate protection to what's going on inside that material, that you don't need to have clean rooms, etc. And so that was the purpose when this work stream started, was looking at how can we do away with clean rooms, and then we've moved on to talk more about closed systems as well. So these are the major bits of legislation that have been published over the years, and we'll start back with ICH Q7 in 2000, 2001 for the FDA. The Annex 2 from the UDRELEX and PICS guidance, and then I've put up on here as well the updated Annex 1 as well, that came out in 2022, because that has got the statement in there about being able to be applied to, or some of it being applied to low bioburden, and we'll come on to that when we talk about drug substance later. So these guys, like I said, started, and this work stream started, the team started publishing back in 2011, and this is one of their first papers, which was about challenging that the clean rooms, why are we using clean rooms, etc. And there was another one in 2013 that backed that up. 2016 they did a response to the Annex 2 and went through, okay, there's some good stuff in there, but there's some bits where we'd sort of recommend a more risk-based approach as well, going forward. We did another paper, which is a really important paper actually, back in 2018, which was this study here where we looked at when we talk about closing systems, we often talk about CIP or SIP, so steam in place, sterilizing place, those type of things, and cleaning place. But this study showed that if we do something as simple as a water flush of a piece of equipment, a pipe or whatever it might be, that we get enough reduction in our bioburden load to be able to call it closed, and for the purposes of a low bioburden process, which is what most of us are operating at in this and in the drug substance world anyway. Then we had a series of papers in 2020 about viral segregation and how much harm can a single drop do, and then we also, towards the end of I think it was in actually 2021, we published a paper in the PDA, which was about the risk-based way of selecting those clean room classifications, so that you don't automatically go to that grade B or grade C, look at grade D and look at unclassified areas as well. So it was all of those that we were looking at, and that's in that paper there. And then last year we published another set of papers, which I'll go into a bit more detail. A paper about filter integrity testing, and then two papers or two chapters of a closure playbook, which we're working on as well. So I'll go on to the deliverables and talk about those ones and the current work we're going to looking at as well. And I think there's some polls going to pop up on your screen as we start talking about this as well, so that you can fill in and answer those polls as well as we go along. So we'll start with the closure playbook, and that's the piece of work that this team has been working on for a number of years, but we've got to the point where we're starting to publish that now, and that's going through and going a little bit further than some of the other things that are out there, like the ISP baseline guide and ASME BPE, and giving you some practical ways of interpreting closed systems and what a closed system is and how to close the system. And we're giving a bit, I suppose, a consensus view of the industry around that. But I've got a little video that hopefully you'll all be able to hear the sound and tell you a little bit more about it as well as we go. Hopefully you can hear that.
The creation of closed systems allows the opportunity for innovation when designing new biologic facilities, as the need for environmental control is reduced. Closed systems technology has been readily available for some time. However, facility design hasn't advanced at the same pace. Closed systems with CNC-based design are quicker and cheaper to build. Fundamentally, they offer greater patient protection and provide a higher quality of product for patients. Companies need to embrace closed systems and make the paradigm shift to new facility designs. BioPhorum has created a definitive guide to designing and operating innovative facilities, demonstrating the use of closed systems as a strategy for making products safer for the patient. BioPhorum's playbook contains risk-based approaches developed around three key areas: process definition, risk assessment, and attributes that identify best practice to meet regulatory standards. The playbook uses globally recognized terminology focused on current standards and regulatory guidance, including the baseline definitions around system closure. This unique roadmap provides tools, direction, and documents on how to implement closed system design for biologic facilities. Join the conversation. www.biophorum.com.
So that hopefully gives you a little bit of information about the closure playbook and what we're trying to do. Like I said, we've already published the introduction, and there should be a link coming up in the chat so that you can go and download that and read that. We've also published a chapter about the regulatory considerations that you need to think about, and our interpretation of that regulatory guidance, and that includes both the Annex 1, the Annex 2, the FDA guidance, some of those ATMP guidance as well, because there's some things in there that people say, oh, that means you can't do this, but when you read the words carefully, we've got a lot of scope, especially using the quality risk management tools that we now use within our industry to do things slightly differently. We've also got the glossary that has been published as well, and key terms, and that's aligned with the ISPE baseline guide, same definitions for what a closed system is, and some of the things have disappeared. So a functionally closed system is no longer in there, or and that briefly exposed is no longer in there. A system is either open or closed, and we functionally close it in different ways, either gamma radiate it at some site otherwhere, or we SIP and steam in place or flush to close it at our own site. So we always functionally close in some manner. And briefly exposed, people got hung up on that, so a quick like that, and that's not what people are talking about there. When we talk about briefly exposed, it's control conditions open operations. So you're opening the process, and in some ways you're controlling that, either by doing it quickly and it's only open for a small amount of time, so proliferation can't happen, and then you filter straight away, or it's something like you have low temperature in there, or you've got extremes of pH or something else that means that you're able to open the system or operate the system open. So there's some of the definitions, and more of that will come out and be explained as we publish the next chapters, which is about operational philosophies, and then a closure risk analysis chapter as well, which is so important. This is probably one of the key things, is doing that upfront closure analysis, working out how you're closing things, where your risks are, where the different inputs, outputs, and everything else are on your systems, and what you need to do to minimize the risk and ensure that you've got a closed system. Then we'll have a chapter later this year, hopefully around facility impacts and some stuff about the design and architectural things that we might want to do. Because CNC, like I said at the beginning, it means a different thing to different people, and it could be from having something simple as a clean room which you just don't monitor for the cleanliness, to something that looks more like, I'd say, an oil refinery in some ways, with the pipes fully exposed and everything else in there, people walking about in just their normal street clothes. Very different from different people to different people, so we'll talk about that. And then the final chapter that we've got is about documentation. It's about talking about and looking at what we've got, what documentation we need to ensure that we document this all correctly, and we can show that our system is closed. So that's the closure playbook that is going to hopefully be finished being published by the end of this year, is our aim, but we'll have another couple of chapters coming out very soon, so look out for that and go and have a look at the chapters that there already are. Another one that we published actually last year was this risk-based approach for filter integrity testing and the requirements for a biologic drug substance manufacturing, and what do we need to do around filter integrity testing. And this came about because of the Annex 1 revision. And the Annex 1 revision said this piece about that you can apply this to low bioburden biologic intermediates, so the API drug substance that we manufacture as biologics, and even when they're not intended to be sterile. And there was lots of sections that talked about filter integrity testing in there, and that word PubSit comes up, which if we talk about Annex 1 in fill finish, we'll spend the rest of the day talking about PubSit. But how does it apply for DS? What do we need to do? What about PubSit? Do we need to start thinking about that in drug substance as well? So we started thinking about this actually before this was published, but and started discussing it and working through the implications of what's happened in Annex 1 for filter integrity testing on closed systems, because we use a lot of filters in drug substance. It's not just that one at the end of the process. We lose a lot throughout the process. So what did we do? We started off, as we often do in BioPhorum, with a survey of the industry, and we looked at the industry and who's doing filter integrity testing. Are they doing it post use? And we had a survey of a sort of I think there was about 40 companies responded in total, and we had all the different filters throughout the process put out and say, do you do it post use? Do you do it if it's in a stainless steel housing, or if it's a cartridge filter? Where and when do you do filter integrity testing? And there was a difference in the industry. yes, in medias, nearly everybody was doing some post-use FIT, but for upstream, downstream, there's a mix. For buffers, there was a different mix as well. So from there, we went and we actually started looking at and we did an FMEA style risk assessment of all the filters in a representative DS manufacturing process. And we looked at both the gas and the liquid filters. And there was over 100 filters that we were looking at, and we looked at the failing modes that you could get and whether putting an FIT gave you a benefit or didn't give you a benefit. And the reason why I'm talking about this as well is because we're presenting this at a number of conferences, and we've actually got one today at the ISP biotech conference in Boston. Mark Peltier is presenting on our behalf around this topic and this paper. But we looked at all of those. And when we looked at that FMEA, it became clear to us that we're actually asking the same questions over and over again. And we were able to simplify it down into some kind of a decision tree. And this is what we'd recommend people doing is working through that FMEA for your product, your process, and your facility. But you probably can get to some kind of decision tree similar to this. And there are times when we'd say, no, you don't need to do an FIT. It doesn't add any value. You've got a bioreactor there. It's the best way of telling whether you've got contamination, and that'll tell you probably quicker than any FIT testing will do, and whether you've got any issue with that filter or not. So there's that. But then there may be cases where actually the bioreactor, we really want to protect it, and we've gone quite a long time. So we might want to do some FIT pre-use and test it pre-use. And then there's others where we'll be filtering into a whole tank or something else where it makes sense again to do an FIT post-use. So there's different ones there that we look through. Follow the decision tree, see if it's the same for you. And what we found was this way of us doing this in this paper backed up an awful lot of what other people were already doing in their companies and had already got in place. So we were fortunate in that way, and people are using this paper as well as backup to their own procedures. So that was the risk-based approach to filter integrity testing. The next thing that we're looking at, and we've been looking at, and I said there was a series of papers about viral segregation, about sort of closed systems helping with that viral segregation and that viral clearance type stuff. We've got another paper that we're working on at the moment, and using those layers of protection that we put in place to protect our systems from contamination. And the ultimate one is that closed system, but what about those other layers of protection in place? Are they giving us a false sense of security? Does that wall that we often put in place, does that actually give us any benefit? Are we seeing anything, or is it the fact that we change our gloves that's the bit that's given us the better benefit? So we're challenging some of those layers of protection, and we're going to publish a paper at some point around layers of protection and viral segregation, and showing that we're good as an industry. We're good around these things, but are the things that we're overcomplicating it? Are we doing too much at times, or are we not doing enough in other places? Are we missing the bit where we really need to focus our energies? So that's that one. The other thing that always comes up, so there's two things that come up whenever we go and present at conferences, that like Mark's doing and Richard and Lars and a few of our other people on our team have done over the years, and we do it frequently presenting at conferences, is around leaks. What about if you get a leak or a breach? So we started a sub-team talking about that, and that moves on to where we are going as the future and what we're looking at for the future in this area. So the first one is around leaks, and it's becoming clear that this is probably going to be a series of papers, probably following and doing something on some kind of decision tree like this, or a flow path that we have around leaks, around looking at that design, looking at that installation of our systems, but looking at what happens if you do. How do we detect leaks? Are we doing some kind of pressure sensors? Or are we relying on people going around with a soap solution and looking for bubbles? So there's different ones that we've got. And then what do we do when we see a leak? What is our immediate response? Do we always raise a deviation? Do we do a root cause analysis? And then how about repairing those leaks? So the first paper will probably be around leak detection and that immediate response, probably something about a preemptive response as well, because we can look at the system and say, if the leak happens there, what would we do? What is the impact of a leak happening there? If a leak happened there, is it different? And we can look at that in a model system. So that's what we're starting to look at. But we're also doing as we always do, sharing case studies, sharing best practice, and we'll do some kind of survey about the industry as well, what's happening. The other topic that's coming out is validation and qualification. That's the other question that we always get asked. What about validation and qualification? And some of that will come out in the documentation chapter in the closure playbook, but also, is the stuff that's published there at the moment, is it enough? Have we got enough information in there? And so we're going to look and pull together probably something around what's already out there, point people and signpost people to the correct publications and the correct pieces of guidance so that tell you what to do. But then is there some gaps, and try and fill those gaps around validation, qualification, say a way of doing it maybe, and it's probably some kind of matrix model that we'd have to put in place, and there's a lot of documentation that you have to have, but let's have a look at that. And that's the piece of work that we'll start once we've got that closure playbook all written and complete. We'll start something on this one as well. And then the other thing that came out of a face-to-face meeting that we just had recently is that we've explained these ideas, and we've changed. We in BioPhorum and within the closed systems team, we're very intimate with them and we understand it really well. But when we explain it to other people, the great big topics, even like just a definition of what a closed system is, it's quite complex. So we need to explain some of these ideas, break them down into smaller chunks, and make sure that we carry on with our conference presentations, do some webinars, podcasts, animations like that video that I just showed, or simple training and instructional videos as well. Get some different smaller messages out there to people. So that's the other thing that we're planning to do as well. But it's all about having that conversation. That's where you get the value from this, is being part of the conversation. And I've talked about what we're doing and everything else, but I haven't told you yet why and some things about the value. So why do we want to do closed systems in the first place? And a bigger is it's for patient safety. Closed systems, they'll reduce the likelihood of you getting those regulatory inspections because you've got a closed system. The environment doesn't matter on your process. The other thing, it reduces the cost of your facility. It reduces the complexity of your facility if it's put in place in the right way. Your product quality should increase. You'll have less contamination incidences, and the risk of that contamination comes down. And if you look at the closure playbook, it should give you those tools to implement closure, and realize that return on investment. And in one of our early papers, we looked at some of this around cost savings and things. And even just looking at CapEx, a grade B room at that time was $200 to $400 per meter cubed. If you go to CNC or a non-classified, it was around 50. So you've got that reduction in that CapEx and making them. But the bigger savings come in your OPEX, your operational, because you get a reduction in energy use. You remove the need for environmental monitoring. You need the need for gowning or people having to change gowns between areas. You'll gown up once maybe to go into the production area, and that's it. And then there's also those reductions in quality investigations. You don't have so many because you don't get those contaminations, and your HEPA filter maintenance is reduced. So all of these add up to a bigger saving as well for you. But this is some of the things that have been reported in the reductions of contamination risk, of those gowning costs. But a big driver for this, which is becoming even more important for our industry, and if we listen to those high C-suite people within your organizations, sustainability is becoming a buzzword that they want to hear, and that is a big driver for closed systems. We're reducing that energy consumption and all of that disposables and everything else that we come along with our normal operating in a clean room. That's all I had to say. I don't know if we've got time for questions. I'll probably just overrun a little bit. So if you've got questions, please put them in the chat, or send them to me via email, or at the end of the call, we should have a little bit of time and we can look at all three sections that we talk about and discuss any questions and anything else you've got. I think that's probably the best point to put it. So with that, I'll hand over to Steve and let him take control of the screen, and he can talk to you about microbial control and all the wonders that there are there as well.
Thank you, Paul. Can you see the presentation? Excellent.
Yes, I can.
As the technology is working. Well, ladies and gentlemen, it is practically a legal requirement that whenever you start talking about microbiology or microbial control, that you start with some interesting facts about microbes. And being a law-abiding citizen, I'm going to follow due course and kick off with a simple thing for you. Maybe you were already aware, but actually every single one of us is surrounded by our own personal cloud, invisible microbes. And it turns out that this cloud is actually almost like a unique bacterial signature. And some research done University of Oregon recently, they showed that they could identify individuals from this bacterial signature even after they'd left a room, which I think is amazing, and the implications of that are both enlightening and a little bit scary. Now, the numbers we're talking about in terms of microbes, quite staggering. Depending on the literature you read, every one of us carries up to 37 trillion microbes, both in and on our bodies. So if you think about the number of people on this call, that would represent over 1,100 trillion microbes that we are hosting amongst this little team. It's quite scary when you think about it. The good news is that, of course, most of these microbes are very benign. We actually need them to survive. But there are a few nasty critters around. Just for your consideration, like me, hopefully you're human. Like me, you use the toilet. But did you know that every time you flush the toilet, you're releasing thousands of tiny little droplets that contain bacteria and viruses, which can actually travel up to 2 meters from the toilet bowl? So my question for you is, where's your toothbrush located? Might want to think about that. Now consider the fact that everybody's favorite toy, the cell phone, or mobile as we call it in the UK, has been shown to contain over 7,000 types of bacteria, and it contains 10 times more bacteria than a toilet seat. And in this case, there have been quite a few studies which show that these phones and our tablets can be harboring some real nasty bacteria, including, as it says here, Streptococcus, Staphylococcus, E. coli, Enterococcus, and even MRSA. So again, the question is this what you want to be putting onto your dining room table? So hopefully that's got you focused on microbes. Let's think about the implications for the industry, biomanufacturing industry. And if we look at the complexity of our processes, our systems, both for upstream and downstream, you can probably start to understand that the challenge surrounding trying to introduce contamination-free processing is immense. But we have to try. It's important. Microbial contamination, or microbes generally, can impact on your product safety, the quality. There are, as you know, strict regulatory requirements about this. It can have an impact on your efficiency. It can have an impact on your shelf life. And if we get any of these things wrong, it can ultimately impact on your brand reputation, your product reliability. So to deal with this, we set up the microbial control workstream. We're really designed to provide what I call an inclusive environment where microbiology experts can come together, they can exchange knowledge, and work together to develop compliant and best-in-class standards and processes, practices that will drive us towards achieving contamination-free processing. We have a vision, and that's simply to become the #1 forum for microbial control experts in our industry to come together to share their knowledge and their best practices. We've got 27 companies involved with over 120 subject matter experts. We meet approximately once a month for an hour and a half, and we have a number of sub-teams running, which I'll describe shortly. The workstream itself has been quite prolific in terms of publications, and I'm bringing some of those up on the screen for you now. They cover quite a wide range of subjects, and they have been used and still and remain as real benchmarks and standards for the industry to use. Many of the publications you've seen are actually a few years old now. And of course, in that time since they were published, our knowledge, our understanding has increased immensely. And we continue to work and develop our understanding around all of these elements. Now, a good example of that, these two pieces of work here. The first paper, which was around low endotoxin recovery, was published back in 2017. That's now been supplemented by a new publication, effectively a second part of this BioPhorum harmonized recovery study, which focused on the use of naturally occurring endotoxins. And I'm pleased to say that that paper has been accepted by the Biologicals Journal. And actually the reviewer was kind enough to say this is a groundbreaking paper and is really challenging some of the preconceptions around the use of naturally occurring endotoxins. So that we're hoping will be published very soon. Another area of continuous concern relates to affinity capture chromatography, or what we call protein A chromatography. And again, here, after an initial paper published back in 2018, we've continued to build on our knowledge and understanding of this topic. If we think about chromatography, protein A chromatography in particular, there are many ways for microbes to ingress into the system, some of which I've listed here. And once they get into the system, what do? We actually feed them with nutrient-rich media from our upstream operations, which helps them proliferate. And they're kind of laughing at us because they know that the sort of chemicals we use can have a very derogatory effect on our resins. So it's a challenging area to work in, but important because we ran a small survey of the workstream members, and every respondent said, yes, we've encountered this issue. We've encountered contamination of our protein A resins, either at sub-alert or alert level, or indeed at actionable levels. Now, most people said, we see this problem in our in-process samples. That's probably not a surprise. Some people, however, even reported having microbial contamination of inbound new resins. When we ask people, okay, of the nasties that you find, of the contamination you find, what are the most problematic organisms? And what they told us was, well, okay, these gram-positive spore-forming bacilli really hurt us because they're remarkably resistant to all the chemical solutions and application recipes that we've tried. So we've taken all this knowledge, all our understanding, and we've created this new publication which focuses on 3 areas. Prevention, of course, is always better than cure. And as Paul alluded to in his presentation around closed systems, risk assessment is always going to be the underlying principle for any preventative approach. But to build on that, we've also provided a very comprehensive list of practical steps that companies and individuals can take to minimize the potential for contamination. But the reality is, whatever we do, however well we do it, there are always going to be instances where contamination occurs. So we've also put in our section around remediation, which is based on a 3-step process. We have focused on chemical cleaning naturally, but we also indicate that the use of pressure and temperature can enhance that cleaning effectiveness. We've given a lot of examples of methods used by the workstream members and how they have successfully remediated contamination of the resin, and examples of things that didn't work. We have included a section on the use of PAA, peracetic acid, and that was based on conversations with the members and conversations with Cytiva, who are one of the leading providers of protein A resins. But as I've said, even successful remediation, what you are effectively doing is using relatively caustic or strong oxidizing agents. And these can have a real impact on your resin and the performance of your resin. So we've added to our paper a section on things that you may want to look at and consider in terms of supporting your resin remediation strategy to ensure that there's no detrimental effect on your product performance. Now, we're very close to publication. We're hoping that will come out in the next couple of weeks. So watch this space. Now, I mentioned risk assessments. In parallel with the Protein A or MAB resin team that's been developing that paper, we have another sub-team that's been specifically focused on generating what we're calling a theoretical case study, which is built around protein A chromatography. And we've actually adopted and adapted the methodology that Paul and his closed systems team have developed. I'm not going to dwell on it too much, but what I can show you is that we've done a fairly comprehensive exercise. And although they're not all listed here, what I can tell you is that for protein A chromatography on its own, we've identified 62 potential causes or methods of contamination. And of course, we're working through that list to understand which of those causes really need to be where we really need to focus mitigation actions. And the idea, of course, is to drive your risk through mitigation and ultimately reduce your risk score as a result of those mitigations. That work is under development, so more to come soon. As I've said, doesn't matter what you do, how you do it, there will always be an element of contamination that creeps into your process. So we've also had a sub-team working on what we call the response to contamination document. Now, they started by generating this process flow. It's a step-by-step guide to how do you respond to contamination and the sorts of things you need to consider. And that was supplemented by, or has been supplemented by, a very extensive document which outlines for every step a lot of detail about how to actually conduct that step. So for example, if you wanted to know about how to do a Gemba walk and what sort of things you need to consider, you would simply click on that button and it would take you to the relevant section of the document. Now, what I'm going to try and do, if it works, is actually show you in real time what it looks like. So here is the under construction piece of work. So again, if you clicked on the Gemba walk, it will take you directly to the relevant section of our supplementary information, and you can check all the good detail around that, and hopefully that will help you form a strategy for conducting a Gemba walk. To get back to the main screen, you simply click on the home button. As I've said, that's under construction. It is due for release in the middle of July, and we're very much looking forward to having that over the line. So, lots of things going on. We talked about the LER paper that's due to be published imminently. We've talked about the MAB resin guidance, which is also due to be published imminently. And in fact, myself and Jakob Sonner Hansen from Fujifilm will be presenting on this paper at the ECA Microbiology Conference in a couple of weeks' time out in Munich. The risk assessment piece and the response to contamination piece also ongoing, very close to completion. Once we've completed these activities, the idea is then the sub-teams that have been working on these will be closed down and new ones will be created to work on the new deliverables. Now they're listed here, but I'm going to skip straight to the next slide to discuss what we're doing in terms of our future direction. So as a result of quite an extensive discussion last year, in fact, we identified two deliverables, which we call our primary deliverable and our secondary deliverable. The primary deliverable is going to be a microbiology process design and controls guidebook, effectively a deconstruction of the entire biomanufacturing process and an identification of where microbial control measures are going to be essential. And as Paul has done for his closed systems playbook, we're going to create it on a chapter by chapter basis. Subtle difference, however. What we're doing in terms of creating this document is to say, okay, we're going to create this stepwise strategy chapters, and we're going to look at them based on the burden to the industry, and we're going to prioritize each element based on that burden of the industry. So it may be that chapter 5 is the first chapter to be written because it's the most pressing need within the industry. One of the beauties of subdividing our process into smaller basic operations is that it helps us identify the right SMEs that we need to be working to resolve these issues, and of course the companies they represent who may have an interest in progression. It also gives us very focused discussions, and it simply creates smaller, faster deliverables for the team. Our secondary deliverable is going to be based around personnel awareness. As you probably know, nearly every contamination event arises because of people. And there's no strong guidance within the industry as to how to increase levels of personnel awareness. So we've defined a 3-chapter approach, which we are looking to start developing in the near future. And on top of that, we will also be creating what's called a self-assessment tool, which will allow individual companies to assess where they are in terms of personnel awareness, and hopefully, as I'll come on to show you briefly, allow them to benchmark where they are, take the survey again in 12 months, 24 months, and see if we've had any significant improvement as a result of adopting these practices. Now, I mentioned sub-teams a couple of times already. We tend to use sub-teams to deliver our objectives, smaller, focused groups. Now, I also told you that we have up to 120 members in the microbial control workstream, and unfortunately, not all of those people have the time. Sometimes they don't even have the expertise to contribute to getting a deliverable over the line. So what we do is we've created what we call a blue sky topic, which is really looking at the future trends and technologies which are going to impact on microbial control operations within a 5 to 10 year time frame. And we started by breaking down the process. In this case, we're looking at the downstream process, and we've identified all the different testing and sampling points within our downstream process. This is not comprehensive, by the way, it's just indicative. And what we're then doing is we're actually delineating each one of these tests, say what it is, why we're testing it, is it offline, at line, inline, etc. How many samples, how long does it take? What we're trying to do is build up a comprehensive understanding of what we call the total microbial burden which we encounter as we produce biopharmaceuticals. Once we have that total microbial burden, we will understand the current state. Our mission then is to develop what we call the desired state. So if we take the first line on this table as an example, let's say we wanted to say, we want to make this into an online test and we want it to take 30 minutes to complete. How do we do that? What technology do we need? How do we implement it? One of the things, and we've already heard from Paul, the importance of closed systems, and we have to take this into account when we're thinking about the future, because we engaged with the L2s, the senior leaders within drug substance. We asked them their thoughts around closed systems, and you could see here that 84% of respondents believe that closed systems will probably become the de facto standard in the future. And 72% believe the transition will take place within 10 years. So in our thinking about future technologies, we have to take closed systems into account. And of course, what that shows is the synergies that we gain between work streams like microbial control and closed systems and Annex 1. And we work very hard to ensure that those synergies are realized. And we take our L2s along the journey as well. We don't just say, hey, this is what we're going to do, and then we come back in 12 months and say that's what it looks like. We try and take them with us on that journey, and they're a very valuable source of information. So what I'll flash up here, you don't need to read this specifically, but this is input we receive from the L2s about how we shape and what they're looking for in terms of this blue sky topic. So two key points, really: the synergy between work streams that needs to be utilized and exploited, and the expertise and knowledge of our L2 community will always be accounted for in what we do. Now, one of the problems we have, or can have within BioPhorum, is how do you measure value? How do you measure the value of what you've done, what you've achieved? And one of the options we're looking at the moment, pending, again, discussions with our L2s, is the creation of what we call a contamination event rate survey. Now, this will be focused on low bioburden drug substance manufacturing, and we know from talking to our members that 90% of companies are using either mammalian or microbial platforms. So we're focusing in on those areas. And what we're looking to do is establish some metrics which will allow us to quantitatively measure the value derived from implementing deliverables in the future. It'll also allow companies to do some inter and intra company benchmarking. Now, we need to think carefully about the target groups that we're going for here, and we need to think very carefully about the key performance indicators. But what we are clear on is that we want to use segmentation to target responses from specific groups of experts. There's nothing worse than getting a 100-question survey and not being able to answer half of it because you're not an expert in that particular field. So by using segmentation, we're going to target people to answer specific questions based on their specific expertise. And we also recognize the need to test and refine the survey by running a couple of small pilots, taking that feedback and making that survey absolutely foolproof. And I believe if we do this and it works and we get the information we need, it's really going to be a very, very useful document for the industry going forward. So there's lots going on. We've got lots of things planned. Just finally from me, What I can say about the microbial control workstream is that we have many highly engaged members who are very willing to share their time, their knowledge, and their expertise. And that's really manifested very nicely by the very wide use of what we call Ask BioPhorum. Hopefully most of you are familiar with what that is. It's probably the most active workstream in terms of its use of Ask BioPhorum. And every single one of these questions that you can see listed here has elicited responses from other members of the team, typically in the region of between 5 and 10 responses from as many companies. So we've done a lot this year. We have an ambitious program for 2024 and beyond, and we want to continue to deliver on our mission. If anything I've said has resonated with you, please feel free to get in touch. And with that, I'm going to hand back to my colleague, Paul.
Hello, welcome back again. I'm going to now talk about Annex 1, Applied in Drug Substance. And like I alluded to earlier, this workstream actually was a reactive workstream, and we started this workstream just over 2 years ago in response to some feedback and stuff that we were getting back from our members along the way about the need for some discussions around Annex 1 applied in DS. And like all of our work streams, again, we have a charter, and there's the charter. I won't go into and dwell on that too much, but this all started because the Annex 1, when it was being updated, it had this statement in there, and it said that basically that it provides a lot of guidance in Annex 1, and some of that guidance around contamination control, design of premises, clean room classification, qualification, validation, monitoring, personal gowning, could be help and support manufacture of products that are not intended to be sterile. So Annex 1 is all about sterile products, but some of the stuff could be appropriate for non-sterile products. And it draws out particularly liquids, creams, ointments, and low bioburden biologic intermediates. So low bioburden biologic drug substance, the stuff that we make and talk about all the time within the DS forum. And when the people saw that, they got all worried and all concerned that we were going to have to hold the Annex 2 areas to these guidance around sterility and everything else that's in Annex 1. Thankfully, there was this extra statement and this extra sentence that was added and said that basically when you're going to use the guidance in Annex 1 in non-sterile areas, you should document which principles and you're going to apply, and then acknowledge and show how you're going to show that you're in compliance with that principle as well. So there was an extra bit which made it a little bit more clarified things a bit. But we were looking at these things and these principles that we're looking at, and because microbial, as we've just heard from Steve, is so important, particulates, pyrogens, endotoxins, they're all important for us in those low bioburden drug substance manufacturing. So that's what we started, a team just discussing it and sharing and understanding what people were doing about Annex 1 in an Annex 2 environment. And Annex 2 was always referred to Annex 1 because Annex 1 is the only place that you've got those classified rooms and information about clean room classes, etc. But what did we start looking at? We started looking at, and the list is there. It's the list that was highlighted earlier around clean room classifications, operations grade, contamination control strategy, design, apprentice, environmental monitoring, and then that filter integrity testing, which we took into the closed systems and did something about there. There's also other areas that we may want to look at in the future as well, and we have a list of them, and we've got several others that we've spoken about on the calls, because we've been having calls every month and discussing this. What are people's hot topics? What are the things that are concerning people at the moment? And that's where the real value is in BioPhorum, is being on that calls, being able to ask the question of your peers and other subject matter experts, find out what's going on in the industry, what are your concerns, what are your problems, and how are you solving them, sharing that expertise. But then we write the papers as well. And the first sub-team that we set up was looking at operations and grades, and that's because there was no clear-cut definition of or guidance for drug substance. There's Annex 1, Annex 2 refers to Annex 1. Annex 1 talks a lot about those high class for glide areas, but the grade C and grade D have a lot less information in them. And open and closed is really a key parameter as well. And if you've got a closed system, as we've already said, potentially you don't need it in a classified area. You could have it in a non-classified space, and so that changes things yet again. But we've still got UDRELEX there that tells us what we should have for GMP and the other GMP guidances from the FDA, etc. So we've still got to comply with those. But then what about CNC? Should we say something about that? That's not in any guidance anywhere. Like I said, each company defines their own guidance. Do we need to strictly follow those clean room cascades? Do we need to go from grade D to grade C to grade B to grade A, or can we go from grade C to grade A, or grade D to AA, or even non-classified and have a grade A isolator in there? Let's talk about that and have a discussion. And there's other conversations that we've had along the way. What if you're refurbing a new facility, an old facility? What if you're building a new facility? There's differences as well. So we're going to write a paper, or we've written a paper already. We're just going through changing the order to make the paper flow nicely. And then that paper will get published later this year once we've got that flow sorted. And it'll give some answers to some of these questions, hopefully, or some ideas about how you may answer these questions in your particular area. We then had another sub-team looking at environmental monitoring. And like I said, Annex 2 is always referred to Annex 1 for clean room classified areas and clean room classification, but it's also by default referred to it for the environmental monitoring as well. So we took the section 9 of Annex 1, which all talks about environmental monitoring, and we did a gap analysis on that, and then we challenged ourselves and said, okay, does this section apply in a DS? Is it different to DP? Do we do something different? And looked at that risk-based approach and of solutions about that, and said for a lot of the areas, yeah, it's fine, it applies. But there's quite a few as well where we'll do something different in DS. We'll take a slightly different risk-based approach around it. And we justified that and explained why we do something differently. And that paper is just going through its final legal reviews at the moment by our member companies who have written that. And that should come out very soon. It emphasizes the importance of quality risk management and contamination control, and tailoring that environmental monitoring to your specific product, your specific process, and your site needs as well. So, and I suppose building in there your company's risk tolerance as well in there as well. So all those different things that you can look at within environmental monitoring. The other area, which was I believe anyway, and I know quite a lot of other people do, is around contamination control strategy and how good that bit in the Annex 1 is around there, and that's a great piece of work that I think a lot of companies are adopting. But it was new when we started talking about this. It wasn't something people had done before. We had contamination control, but it was spread all over the place. We didn't have it pulled together in one document and one specific place. So we've talked about how people are doing that, what people were doing about that, how they're bringing in things like the closure definitions, what about KPIs and that effectiveness check? What does best in class look like for a CCS document? Are people using a computer system? Are people doing it on paper? What people are doing for that as well? And are there differences between a DS facilities need and a drug product systems need? And over those discussions, we had case studies from different members. We had focused discussions on different topics and lots of other benchmarking as well along the way with surveys that we published internally. And we did all of that, but then we realized that there's been a lot of documents published by the PDA, ECA, and lots of other people around CCS, and we realized that a lot of those wasn't something extra that we'd want to add, but there was around effectiveness review and around that effectiveness check, and who does that? What is it even? Where is it done? When is it done? How frequently is it done? Yearly, bi-yearly, every 6 months, every quarter? What is it? Why are we doing it? And how are we doing it? So we're going to publish and write something around effectiveness review and that effectiveness checks, and publish something around that. It'll probably be the end of the year because this team's just started writing on that. But it's what we're looking at the moment, and trying to answer some of those questions. What does a sort of a minimum standard look like, and what does best in class look like for these things as well? What do we want to do, and where do we want to position it? Because the environmental monitoring paper was getting close to being finished, and I don't like to be sat about, and I don't like the team to be sat about, we started off another sub team looking at gowning as well, and so this is the newest sub team, and it's to look at tackling that challenges around for the DS operations about understanding the requirements of Annex 1 and gowning, particularly for those low classified rooms, so those Class C, Class D rooms, and even non-classified rooms. What do we need to do for gowning? So we ran a benchmarking survey, like I said, most of our things do, and this was we got over 40 responses for this benchmarking survey from 150 questions, so it's a really in-depth survey. It took over an hour for people to fill in this survey, so a lot of questions, and we're still going through analysing those questions and looking at them and pulling it together, but we'll pull it together in some kind of member published document, and this is one where it will be available to our members to start with, and then we might take snippets of it and put it into the paper that we publish as for everybody to see later on. But some of the questions that we ask are things like dedicated shoes. Do you use dedicated shoes in different environments? And as you see, there's different responses, and we split out between EU grades and ISO grades as well, because we have a difference of how people classify their rooms. Not everybody uses those EU grades, not everybody uses ISO grades, so differences, so why we split them out, and there are subtle differences in those two systems as well. So we had that one. We had the other topic that will take up the majority of any conversation if you talk about Annex 1 with the fill finish guys, about clean room socks and single use versus laundered socks. Do you use socks in grade C, grade D rooms? Quite a lot of companies are doing, but then there's also a lot of companies that it's not applicable to them. They don't use clean room socks, so a very different mix in here for drug substance. And if I ask that in fill finish, it would be up there. Everybody's using them for those C and upwards grades. And then we had a question about gloves. Do you use gloves permanently as well? And there was a difference there as well. And as you see, there's a lot of people that use them when we've got those local protected environments where you're doing critical operations. That's when they'll use gloves, but they won't use them in every other area, or they'll don them just for that when they go into that grade C area and above. So there's differences, and we're still going through and analysing that data and looking at that data as we go forward. But we're looking to then do a science-based paper around gowning for those low classified environments, and bringing in some things about closed systems and whether you need gowning and when do you need gowning, and sort of risk-based approach around that. So watch this space, and if you haven't already signed up for the newsletter on BioPhorum.com, sign up on there, and then you'll get first notification of when these papers are published. And like I said, they are freely available on BioPhorum.com, so have a look, sign up, and then you'll see this paper when it is published towards the end of the year. But the key message from everything around Annex 1 was don't just adopt what's written in Annex 1. Don't just raise the bar up to that sterile area. Do a quality risk management, do a risk assessment so that you focus on those important areas, and base it on the protection that your product, process and site needs to ensure patient safety. That's what it's all about. And there are a lot of good things in Annex 1, but there's a lot of things that don't make sense necessarily for us in a low bioburden drug substance manufacturing site to adopt. So do it based on risk, but there's also things about making sure that you document how you interpret guidance as well. Things like closure. How do you, what is a closed system for you? Do you use the BioPhorum ISPE baseline guide definitions, or do you have your own definitions of it as well? But the expansion of Annex 1 beyond drug product makes that risk assessment so important and paramount. And I think that's what's come out of all the discussions that we've had is doing those risk assessments, looking at your site, your product, your process, and basing it upon those things. So watch this space for more papers coming from this team. And like I say, it's been around for a while. If you want to know more, just get in contact, and I can try and answer you and get you involved if needed as well, if you'd like to.